Right, so I thought I'd start with a list of neurodegenerative diseases you have here. They're all grouped together because they're characterized by the presence of abnormal protein deposits. They also group together because there are no mechanism-based therapies for any of them at the moment. Where therapies exist, they are symptomatic, so they treat the symptoms of a disease rather than any sort of cause, and that's basically the sort of drive behind the type of work we and many other people are doing. Today I'm going to talk about 3 of these 9 diseases here in the context of tau protein. So these are Alzheimer's disease, some cases of frontotemporal dementias, and PSP. I'm not going to talk about the other diseases. Now, tau protein is aggregated in many human neurodegenerative diseases. It's probably, as far as individual proteins go, the one that's aggregated in the largest number of cases of disease. And as you probably all know, tau protein in each of these diseases goes from a soluble microtubule bound to a hyperphosphorylated insoluble and filamentous form. And we believe that this pathway, basically, is what one has to try and understand. It doesn't necessarily need to believe that the final filaments are what's doing all the damage. There could be some intermediates that are involved, but I think it's probably fair to say that it's something along this pathway that causes neurodegeneration in a large number of human diseases. Tau protein exists as 6 different isoforms in a normal adult human central nervous system. So there's one tau gene, which is schematically shown up here. And from this one tau gene, you generate 6 different forms of the protein, which differ by the presence or absence of 3 inserts, shown here in red, green, and yellow. I'm not going to say much more about the red and green inserts because we do not know at present what their direct relevance for the neurodegenerative process is. But I'm going to talk quite a lot about the insert here shown in yellow. Depending on the presence or absence of this insert, which is encoded by exon 10 of the tau gene, it's a 31 amino acid sequence. The tau isoforms can be divided into 2 groups, those with 3 repeats each, so they lack this exon 10 insert, and those with 4 repeats each. The repeats are indicated here by R, R1, R2, R3, R4. They're located in the carboxy terminal half of the tau protein, and they are, to a large extent, not exclusively or entirely, but to a large extent responsible for the ability of tau protein to bind to microtubules. And we believe that that's its normal physiological function, at least as far as we understand it. It's also important to know that the same region of the tau protein is what forms the core of the filaments that tau protein forms in the various human neurodegenerative diseases. So one would believe that the normal binding of tau to microtubules and its abnormal aggregation into filaments are 2 mutually exclusive phenomena. In a normal adult human central nervous system, the ratios between the 4-repeat and the 3-repeat tau isoforms are roughly 50-50. So the same amount of 3 and 4-repeat tau is found. And again, in some of the human diseases, this balance is disturbed. And one believes that that's important in leading to disease, especially in diseases where 4-repeat tau, so these 3 isoforms, are present in the filaments. And PSP is obviously one of these diseases, so-called 4-repeat tauopathies. The next slide shows you the typical neuropathological picture of Alzheimer's disease. Unlike the other diseases I had on the slide at the beginning, Alzheimer's disease is characterized by 2 types of abnormal protein deposits, one in the extracellular space, which are the amyloid deposits, and one inside nerve cells, which are the tau deposits. And you probably know that there still is a lot of debate about the relative merits of these 2 types of deposits. It's probably fair to say that based on current understanding, we believe that the events leading to this deposit may be the initiating event in Alzheimer's disease, but it's also clear that the events leading to this type of deposit is what ultimately causes the neurodegeneration. So this may be a sort of initiating event, and this, I would say, would be the sort of executioner phase of the disease. And in terms of potential therapies, I would think that in the long run, one probably will need to be able to do something about both of these types of inserts. The mechanistic connection between these two, and there must be one, is one of the major sort of unsolved mysteries in the field of neurodegenerative diseases in general, and an interesting topic, I think, for future work. This is all light microscopy. Now, if you look by electron microscopy at these types of inclusions, so the tau inclusions, you find that they're made of abnormal filaments. They are the so-called paired helical and straight filaments of Alzheimer's disease, shown here as P and S. The vast majority of the filaments are paired helical, about 80%, about 20% are straight filaments. And this is pathological, so this you would not find in a normal brain. So the aggregation of tau protein leads to these filaments, and this is so the end product of the aggregation process. So this is electron microscopy. If you now take this material, run it on a blot, and immunolabel the bands with a tau antibody, you get this characteristic pattern. Here you have 3 strong bands and 1 weak band. They do not align with the recombinant tau isoforms. So these are the 6 tau isoforms I showed before, expressed in bacteria as a sort of molecular weight markers, if you like. If you take this material here and you dephosphorylate it, it shifts from this into this pattern here, and you can see that now there's a reasonable alignment of these bands with the recombinant tau isoforms. This one runs as a smear because the alkaline phosphatase that's used runs at the same position. So you have 4 strong bands and 2 weak bands of tau, which is the normal banding pattern of tau in the brain, and they align reasonably well with these 6 isoforms. So what this means then is that in the filamentous tau in Alzheimer's disease, first of all 6 tau isoforms are present, and secondly, each of the 6 tau isoforms is hyperphosphorylated. And this is a sort of characteristic of the filamentous tau in Alzheimer's disease. It's also a characteristic of the filamentous tau in normal aging that forms to some extent, and I'll come to that in a minute. The next slide shows sort of what is known about phosphorylation sites in tau from the abnormal filaments in Alzheimer's disease, and you can see there's a very large number of phosphorylation sites that has been identified. It looks like a sort of bulk phosphorylation of the protein, but there are some sites that are clearly more important than others, and I don't have really time today to go into that. But suffice to say is that in the paired helical filaments and straight filaments of Alzheimer's disease, tau is abnormally phosphorylated. And this abnormal phosphorylation of tau is a general characteristic of tau in all filamentous deposits. So you find hyperphosphorylated tau in the filamentous deposits in PSP. You find it in some forms of frontotemporal dementias. You find it in corticobasal degeneration. So it's a general characteristic, and we believe that it's probably an early phenomenon, that it probably precedes the aggregation of tau into filaments, and as such could be a therapeutic target, either to reduce the phosphorylation with protein kinase inhibitors or to increase the dephosphorylation by activating protein phosphatases. Now, as a function of age, in the human brain, tau deposits form in a predictable manner. This is the basis of the so-called Bragg staging of tau pathology, in such a way that at very early stages, small numbers of tau deposits form in the transentorhinal cortex here, where they then, from where they sort of seem to spread, if I can use this somewhat loaded term, into the hippocampus and parts of the cerebral cortex, and then from there they appear, after that they appear in the neocortex in large amounts. People at this stage have no cognitive impairment. Some people here have some cognitive impairment. Here people clinically have Alzheimer's disease. So this forms as a function of age in a predictable manner. The tau is made of all 6 tau isoforms, as I said before, and this may have something to do with the fact that Alzheimer's disease is an age-related disorder, so that its incidence increases with age, and it is very common. These are aggregated tau deposits in these cases, are made of filamentous hyperphosphorylated protein. In more recent work, Brack and Deltridici have described an additional stage, which is probably an even earlier stage, which is based on the presence of hyperphosphorylated tau, but not necessarily aggregated tau, now in the locus coeruleus, so a subcortical region. Again, all 6 tau isoforms appear to be involved in this process. So this is Alzheimer's disease and normal aging as far as tau goes. In addition to this, we have a number of diseases where filamentous deposits made of tau protein are found. So these are arteriophilic grain disease, they're Pick's disease, PSP, and CBD. And as you know, some of these are dementing disorders, whereas others are primarily movement disorders. And the question arises, if one says that in all these diseases one finds filamentous tau deposits, the question arises, one has to ask, why are they not all the same disease? If they all are characterized by filamentous tau deposits, in principle, could all be one disease. And the reason why they're different diseases is probably something to do with the fact that the filamentous tau deposits appear in different parts of the brain, and to some extent in different parts, in different cells in the brain in these various diseases. So in Alzheimer's disease, the filamentous tau deposits are primarily neuronal. In arteriophilic grain disease, one finds them in neurons and in glial cells. Pick's disease, predominantly neuronal. And these 2 diseases, one finds filamentous tau deposits in both nerve cells and glial cells. The question then arises, why should this be? What are the molecular differences in tau or in aggregating tau that leads to different parts of the brain and different types of cells in the brain being affected in these different diseases? And the answer to this question is we don't really know at this point, but there are clearly some molecular differences between some of these diseases. I already said that Alzheimer's disease is characterized by the presence of all 6 tau isoforms in the abnormal filamentous deposits. If you now look in some of these other diseases, you find that in Pick's disease, for instance, only tau isoforms with 3 repeats, or predominantly tau isoforms with 3 repeats, are found in the abnormal filamentous deposits, whereas in these 3 diseases, tau isoforms with 4 repeats are found in the abnormal filamentous deposits. So these would be so-called 3-repeat tauopathies, or a 3-repeat tauopathy, and these would be 4-repeat tauopathies, whereas Alzheimer's disease and Alzheimer's disease and aging are characterized by the presence of all 6 tau isoforms in the deposits. This is reminiscent, doesn't prove anything, but it's reminiscent of the possible existence of conformers of aggregated tau that may differ between these different diseases. And that's a sort of analogy with what has been reported in prion diseases. So you could, based on the presence just of different tau isoforms in the aggregates, you could envisage that this could lead to different types of tau aggregates. But even where the same tau isoforms are present in the aggregates, like in these 3 diseases here, so-called 4-repeat tauopathies, you could envisage that for each of these there are different conformers of aggregated tau that lead to different types of inclusions in different parts of the brain, and therefore to different types of clinical symptoms of disease, and ultimately to the denomination of a different disease entity. This is Western blotting. This is also reflected at the level of tau filament morphologies. So if you look in Alzheimer's disease, I already showed this, paired helical and straight filaments. If you look in Pick's disease, you find these types of filament morphologies. If you look in so-called 4-repeat tauopathies, you find different types of filament morphologies again. So this is again consistent with the existence of different conformers of tau. Now, of course, so far everything I've said is sort of, well, it says tau is present in these diseases. It doesn't tell you anything about the relevance or not of tau aggregation for the neurodegenerative process. And this was clarified and made, I think, proved basically beyond doubt by the study of a rare familial condition called frontotemporal dementia and Parkinsonism linked to chromosome 17. This was in the late 1990s, and we were involved in some of that work. So this is a familial disease that's caused in a dominant manner, and that normally around the age of 50 in people leads to profound personality changes, and these people end up being severely cognitively impaired and then die. Invariably, within their brains, you find abnormal tau deposits. There's a whole variety of these conditions. Some resemble PSP, some resemble CBD, some resemble Pick's disease, some even resemble Alzheimer's disease, both clinically and to some extent neuropathologically, but they all have tau deposits in their brains, and they all have filamentous tau deposits in their brains. And again, the filament morphologies vary to some extent between different families. We now know that this disease is caused by mutations in the tau gene. This is a sort of picture, current picture. There are about 45 different mutations known in causing FTDP-17, called now FTDP-17T. They are heavily concentrated in exons 9, 10, 11, and 12. Now, exons 9 to 12 of tau encode for the 4 repeats of tau in the 4-repeat isoform. And you can see that, for instance, exon 10, which is the one that gives you the second repeat and leads to the difference between 3 and 4 repeat tau isoforms, many mutations are present in that exon. Other mutations that flank exon 10 are intronic mutations, and depending on their presence, an abnormal ratio between 3 and 4 repeat tau isoforms forms in these people that lead to a predominance of 4 repeat tau over 3 repeat tau, and presumably that causes disease. And in these people, only 4 repeat tau is present in the abnormal filaments of the disease, similar to what has been described in PSP and CBD. So about 40% of these mutations are caused by abnormalities at the RNA level. 60% of these mutations are caused by abnormalities at the protein level. And most of these are single amino acid changes that lead to impairment of the ability of tau to interact with microtubules. So there are mutations that act at the protein level, there are mutations that act at the RNA level. In all cases, you have filamentous tau deposits that form within nerve cells and sometimes also within glial cells. And clinically, they all lead to frontotemporal dementia and Parkinsonism linked to chromosome 17. Some of them have a stronger dementia component, others have a stronger Parkinsonian component, again, depending on where in the brain the deposits form. This is obviously a genetic condition. This is about as genetic as you can get. As a single amino acid change in a protein causes disease within sort of prevalence that's close to 100% in individual families. The next condition, this one here, chronic traumatic encephalopathy, you could argue in a way is about as environmental as you can get. These are people that have repeated head injuries and rather non-specific sort of phenomenon, if you like. But at the pathological level, it is very specific in the sense that these people develop filamentous tau deposits in particular brain regions and associated symptoms. So this just to illustrate that as far as the tau deposits go, they sort of can span the whole spectrum from genetic to environmental disease. And again, I think as far as these so-called prion-like phenomena go, this may be quite interesting, because you could argue that in these people what may happen is that at some point in a very small number of nerve cells, filamentous tau deposits form, and then over a long period of time they spread from there to other parts of the brain. And I'll come now to experimental studies investigating this phenomenon, but before I do that, I must introduce a transgenic mouse line that we've produced that has played a large role in these studies, which is a line of mice that expresses one human tau isoform under the control of the mouse Thy1 promoter, which leads to largely neuron-specific expression. And these mice in the tau carry the P301S mutation, which is one of the mutations that in humans causes frontotemporal dementia and Parkinsonism linked to chromosome 17. And if you produce these mice, first of all, at the age of 4 or 5 months, the mice become severely ill. They develop a very severe paraparesis, and they can't feed anymore. And in the end, they have to be culled. If you look histologically in the brain, using a phosphorylation-dependent tau antibody, which is what we've done here, you find tau inclusions all throughout the central nervous system. Down here is the spinal motor neurons in the spinal cord. Everything you see here is basically pathological. If you did this in a normal mouse, you would see at best some neuropil staining, but nothing like this. This is light microscopy. By electron microscopy, you see abnormal filaments. This is brainstem. Oh, sorry, this is cortex, brainstem, and spinal cord. This is again a motor neuron in the spinal cord. If you look at higher magnification in the cytoplasm, you find abnormal filaments. And these filaments are tau filaments because they're decorated, you can see here, with these gold particles lining the filaments by a phosphorylation-dependent tau antibody. If you look at the survival of nerve cells, in this case in spinal cord, but you can find similar results in other parts of the brain, you get a massive loss of motor neurons. Here's about a 50% loss of motor neurons in the spinal cord in these animals. So at a molecular and cellular level, these mice reproduce basically what you find in the human diseases. They do not have frontotemporal dementia, though I'm not saying that, probably because we're using the Thy1 promoter, which is an artificial promoter in this situation. But at the molecular and cellular level, you get hyperphosphorylated tau filaments, and neurodegeneration. So this is one line of mice. Now, the second line of mice that we produced some years ago expresses wild-type tau under the same control of the same Thy1 promoter. So this is one tau isoform here, which has no mutation in it. It also carries the 2 amino terminal inserts, which this one does not carry. And this proved useful experimentally, although I don't think it's relevant for the results themselves. Unlike this line of mice, which carries P301S mutation. As I already showed, this line of mice develop massive accumulation of tau in the brain, of aggregated tau in the brain, and develop neurodegeneration. This is exemplified here by this silver stain called the Galliage silver stain. You can see there's a lot of staining. In this line of mice here, you never ever get any silver positivity in the brain over the whole lifespan of the mouse. You have some staining for hyperphosphorylated tau, indicated here by the 88 antibody in this line of mice, but that's about where it stops. And for many years, these mice were sort of interesting but not terribly useful, because one didn't really know what they were a model for, because there was no human disease that had these characteristics. But in these experimental studies, they became useful. So the experiment was very simple initially, which was to take brain extract from this mice here and inject it into these mice and see whether over time silver positivity appeared here that you normally never see in these ALZ17 mouse line. And the answer is that you can see that. This is illustrated here. If you look at the panel on the right-hand side here, this is the ALZ17 line injected with a brain extract from the P301S line, and you now get silver positive staining here in the hippocampus, which is also positive with this 88 antibody, and 8100 is another phosphorylation dependent antibody for tau. You have some 88 positive staining in these mice, as I showed before, in the ALZ17 line, but no filamentous or aggregated tau and no neurodegeneration. And if you do sort of various controls, you don't see the same phenomenon. So this sort of showed that you could call this transmit the aggregation of tau from one mouse line to another through injection of brain extract from a mouse line that had tau filaments. Various experiments that I don't have time to go into show that what aggregates here is the tau that's produced in the transgenic mouse line. So it's the wild type tau without the mutation. So it's not just the stuff we've injected that somehow survived. That's very, very clear. This is, of course, crude brain extract, and the question is, what is it in the extract that does this? At a first approximation, we could show that, first of all, this phenomenon is tau dependent. So if you immunodeplete tau from the brain extract, you have no aggregation. If you separate the tau into soluble and insoluble tau, it's very clearly the insoluble tau that carries the activity. It's not the soluble hyperphosphorylated tau. So there's something in the insoluble fraction that carries this. At the moment, we don't know exactly what this is, but this is obviously an important question for the future. In more recent experiments, we could also show that now instead of using crude brain extract, you take recombinant tau with the P301S mutation, you assemble it into filaments with heparin, which is a method we described some years ago. So this is now aggregated mutant human tau made in bacteria, but not phosphorylated, and you inject that into, in this case, the P301S mouse line at a time when it has no inclusions. Over time, you get, after a month in this case already, you get a large number of filamentous tau inclusions formed following the injection of the recombinant aggregated protein. So I think this strongly suggests, first of all, that it's entirely tau dependent, that it's probably dependent on the presence of tau aggregates, but what sort of aggregates we don't know. But this probably will allow us to separate different forms of tau aggregates in future and inject them separately and find this out. Now, if you looked at the different types of inclusions you find following injection, now from the P301S extract into the ALZ17 line, there are three types of inclusion types you find. They are neurofibrillary tangle-like structures, they are neuropil threads, and they're also coiled bodies in oligodendrocytes. If you inject, and all the work I'm showing was following injection of brain extract into the hippocampus and the cerebral cortex. And if you separate these, in this case in the hippocampus, into the three components, you find that the neuropil threads and the coiled bodies over time form in the far larger numbers than the neurofibrillary tangles. So in these experiments, animals are injected when they're 3 months old, and we follow this for 15 months after the injection. So the animals are 18 months old at the final time point. You can also see that in the hippocampus, more of these structures form than in the cerebral cortex following injection, and that's something we've always seen in all experiments. The next question was, does this spread beyond the injection site? So the injection sites, as I said, are the cerebral cortex and the hippocampus here, and the answer is yes. It looks like it's spreading both at the injection level, but then also anteriorly and posteriorly. And this is illustrated here at the level of injection, anterior and posterior to the injection site. The longest distance from the injection site, as far as we could tell, was following injection and finding aggregation in the hypothalamus, which was something like 4 millimeters away in the mouse brain, which is quite a long distance. So this is all experiments injecting either recombinantly aggregated tau or P301S tau into the ALZ17 line. The next question, obviously, is what happens if you use human brain extracts and inject those into the ALZ17 line. And this is something we've been doing quite extensively over the past few years. And I'll just show some of these experiments. So the first one is to inject Alzheimer's disease brain extract or brain extract from people with a so-called tangle-only dementia into the ALZ17 line. Alzheimer's disease brain extract obviously contains both aggregated tau and aggregated A beta. In order to distinguish or exclude a possible A beta mediated effect, we use these neurofibril-tangle-only dementia cases as well, who have a lot of aggregated tau in their brains, but no aggregated A beta. You inject this into the ALZ17 line, and after about a year or so following the injection, you get silver-positive material formed. Now, this material is obviously made of all 6 tau isoforms in an aggregated state. When we inject it into a mouse line that only contains 4-repeat wild-type tau as a transgene. And so the morphologies of these aggregates that are formed here, I guess, are not really interpretable, but it clearly works experimentally, so you get aggregation. What happens if you take, this is just another experiment using the senile dementia tangle-only extract, and shows you another picture of stained cells, in this case with the 8100 antibody, which is characteristic of hyperphosphorylated tau in a filamentous state, in this case in the hippocampus. So in this ALZ17 line, as I said earlier, you never ever see anything like this unless you inject these types of extracts. The next experiment now is to inject Pick's disease extract into the ALZ17 line. Now, Pick's disease contains aggregated tau, but the aggregated tau is of the 3-repeat form, and the transgenic line ALZ17 expresses 4-repeat tau. So the question is, what happens if you take aggregated 3-repeat tau, inject it into a 4-repeat tau expressing mouse? And the answer is nothing. So you don't get any aggregation of ALZ17 4-repeat soluble tau if you inject a Pick's disease extract, which I think would probably suggest that 3-repeat aggregated tau can somehow not form a sort of template, if you like, for the aggregation of soluble 4-repeat tau. In a way, that's also not too surprising. The more interesting experiment then more interesting experiments in this context would be to take brain extracts from human diseases where 4-repeat tau aggregates and inject them into the ALZ17 line where soluble 4-repeat tau is being expressed. So this is argiophilic brain disease, progressive supranuclear palsy, and corticobasal degeneration. And I'll now turn to experiments using these types of extracts. Here's an experiment where we injected argiophilic brain disease extract into the ALZ17 line. Now, argiophilic brain disease in humans has a distinct advantage as far as tau aggregation goes, that the morphology of the tau aggregate is very distinctive. So you have these little dots formed, and this should be very easy to identify in a transgenic mouse line. And sure enough, when we did these experiments, injecting this extract into the ALZ17 line. Now, in the ALZ17 line here in the hippocampus, after a year, I think this was following injection, you get dots that are silver positive and are very reminiscent of the material that was present in the human brain that was injected. So this is, I think, very strongly reminiscent of the phenomenon of, well, supporting the existence of possible distinct conformers of tau protein in these different diseases. And this is not dissimilar of what in the prion field are called strains, in the sense that you have something in the extract here, you inject it into the mouse that normally only has soluble tau, and you reproduce to a surprisingly accurate degree in the mouse what was present in the human brain. So this is argiophilic brain disease. The next one now is progressive supranuclear palsy. So you take an extract from a PSP, I think it was striatum, and you inject it into the ALZ17 line. You also get aggregates forming in nerve cells. You can see here in the hippocampal formation. So everything that's black here is Galliard silver positive, something you never ever see in this mouse line unless you inject and something like this. If you look now at the level of nerve cells, you can see in the hippocampus, you can see nerve cell staining. This is pathological. This is stained with the 8100 antibody and is probably reminiscent of tau filaments forming. The morphologies otherwise are probably hard to distinguish between what you see here and what you see in several of these other diseases following injection. But the distinctive feature that we see in reproducibly using different extracts from different brains, individuals who died with PSP, is that we see in the astrocytes, we see these types of abnormalities, which are similar to the astrocytic changes that you see in human PSP. So this is PSP. If you now take corticobasal degeneration, you inject brain extract into the ALZ17 line. Again, you get a lot of silver-positive material forming in the hippocampal formation. If you look at nerve cell staining, you have some here in the hippocampus, but these are silver-positive cells. Again, the morphologies probably don't tell you that much, but then again, if you look at the glial cell level, you see something that's sort of reminiscent, I guess, of an astrocytic plaque, which is, in humans, characteristic of corticobasal degeneration. So it would sort of suggest that there may be, even between PSP and CBD in this experimental setting, there may be differences that one can detect at the histological level and that are reminiscent, at least, of the differences that you see in the human pathologies. So all these experiments were done injecting human brain extracts into mice transgenic for wild-type human tau. Next question is, what happens if you inject brain extracts from human diseases into normal mice? And the answer is, it also works, although overall it works less well than if you inject it into the ALZ17 line. And one has to remember that here we're injecting human brain extracts containing aggregated human tau into mice, so there's a species barrier because these mice have no human tau whatsoever. And it remains to be seen whether that's the underlying basis of the differences. So this is using C57 black 6 mice injected with Alzheimer's disease brain extract. C57 black 6 mice injected with argyrophilic grain disease extract, all in the hippocampus, and with PSP extract, again in the hippocampus. And you can see there is some staining, mostly of neuropil threads in these, under these conditions. How the pathology spreads from the injection sites in both ALZ17 mice and normal mice following injection of extracts from these different diseases is something that we're still working on, so I don't really know the answer to that. So the interesting question is, for instance, do the aggregates following injection of the ALZ17 mice go to different parts of the brain depending on what type of brain extract from what disease was injected. So this is sort of what we believe is going on, is that the aggregation happens in a localized fashion at an early stage. We also believe that this may be something that's very common. This may be something that's happening in all of us all the time, but there may be mechanisms that we have to remove this material at an early stage. In some people, the material may not be removed or may not be removed quickly enough, and once it has started to aggregate, it then, given enough time, may spread throughout the brain over a long period of time. So the initial event may be stochastic in the sporadic disease cases. So it may, in the end, be impossible to predict who's going to get one of these diseases. But the following events after aggregation, successful aggregation, may be more deterministic and lead to the spreading of pathology throughout the brain. And this is just to illustrate, what may be happening. So there must be mechanisms by which aggregated tau, which is indicated by this, is taken up by nerve cells, transported probably through the synaptic terminals, taken up by neighboring cells, and over a long period of time spread through the brain like this. There also may be mechanisms, this is something we're working on at the moment, by which nerve cells are able to defend themselves against these aggregates. So you could imagine that these are like foreign bodies in the cytoplasm of a nerve cell. It's like a bacterium or a virus being present in the cytoplasm of a nerve cell. And cells may have mechanisms by which to get rid of this material and defend themselves successfully against it. And I think that's an important area for future research. So all this was done in transgenic mouse brains. We also developed, and other people as well, assays by which one can study similar phenomena in cultured cells, I think, to get to the mechanisms underlying this, which is obviously important. So here what we do is we just transfect tau into kidney cells, either wild type or mutant, and then we add a seed, the seed being brain extract, for instance, from the P301S transgenic mice to the outside, and over time you can find aggregated tau forming inside these cells after you've added the seed, shown here for the wild type tau and here for the P301S mutant tau. You do not see this phenomenon at all if you don't add a seed or if you don't express tau inside the cells. And I think this is my last data slide. You also get the same phenomenon if you add recombinant tau as a seed. So here we're comparing recombinant aggregated tau used as a seed and brain extract used as a seed. But overall, and I think this has been observed by other people for other proteins as well, like alpha-synuclein, prion protein, and SOD1, is that the brain extract, as far in terms of quantity of aggregated material, the brain extract always works much better than the recombinant protein. So if you add recombinant aggregated protein to get the same effect as with the brain extract, you have to add much more of the recombinant aggregated protein than of the aggregates you have present in the brain extract. So there may be something in the brain extract, in addition to the aggregated protein, that facilitates these phenomena, cofactors or something like that. And I think that's an important area for the future as well. And finally, I would like to thank my colleagues. So we've done all this work. First, in the MRC Laboratory of Molecular Biology, where I work, Maria in the Center of Brain Repair, and then Florence Clavaguera and Marcus Tolnay in Basel, who we've done lots of these experiments with. Thank you very much.
Okay, we're on. So two quick questions. One, the ALZ17 mice, the promoter for that was what again?
Thy1.
Thy1. And so is Thy1 expressed in the non-neuronal cells? Does Thy1 express? In other words, you see
yes.
tau accumulation in non-neuronal cells, and the expression was mainly neuronal.
Yes, that's true. I mean, that's the fact that we see these coiled bodies following injection, I think is surprising for that reason. Now, what we cannot exclude is that you have low levels of expression in non-neuronal cells. In principle, the bulk of the expression using the Thy1 promoter is in neuronal cells.
Right. So that makes it even more surprising. I think that's interesting. And then the other quick question is, so you're putting the extracts onto the cells.
Yeah.
Do you see evidence for strains yet there? So the dots, for example, in the argyrophilic brain disease that you saw.
In terms of the culture experiments, we haven't done much at all with using human brain extract so far, so I don't know. Yeah, I mean, that's interesting to do.
Hey, Michel.
Thank you.
Michel over here. I wanted to ask you, the tufted astrocyte pathology that you see, is that exclusive to the PSP injected?
So far, yes.
So you don't see any of those with AD or anything like that?
No.
And how frequent are they? Like, do you see tangles and tufted astrocytes?
Yeah,
right.
So I would say probably about, you see many more tangles. I would say probably about 1 in 20, something like that, roughly speaking.
Thanks.
So that's really great work, and it seems I'm sure you saw Virginia Lee's paper this week for PD. So this really seems to be gaining ground in many diseases. So given that, what do you think the implications are from a therapeutic perspective, both for the target and actually how we do trials?
Yes. So first of all, I would say this is probably only relevant, or most relevant, to the sporadic forms of disease, because you could argue that in the familial cases where you have mutations, you have them in all the cells, so you don't need this phenomenon. You may still operate, but you may not need it as such. So first thing, because many of the trials now are being done in familial cases, especially in Alzheimer's disease. Secondly, as far as the sporadic cases go, it's hard to tell. You know, it depends if this phenomenon takes off at a very, very early stage and then sort of goes on relentlessly. You may be able to interfere with the progression of the phenomenon, but I would say once it has taken off, for instance, using, I don't know, anti-aggregation inhibitors, for instance, may be too late at that point. Using antibodies, for instance, may not be too late, because you could argue that this progression, even for tau alpha-synuclein, unless they're present in exosomes or some compartment, if they go from cell to cell, they have to be extracellular at some point, and you could think that an antibody could mop them up. So that might still work. But it depends a bit on how early this phenomenon starts. You know, if it's 10 years before symptoms, 20, 30, 40, we don't know. Hopefully at some point we will have, or soon we will have some sort of PET label or so that identifies tau and alpha-synuclein aggregates specifically, and then we will be in a position to do that.
Just a small question. The human brain extracts were from end-stage Alzheimer's disease patients. Would you speculate on, you know, if you're able to get your hands on brain extracts from somebody with amnestic MCI who died from a car accident?
Yes, I think if you had somebody like that, depends which brain region you would use to some extent, I guess. But I would say, let's say if you took the hippocampus from somebody like that, I think you would have aggregates in there too. So I think that would still work. The question is, though, what it is in the aggregate that does this. We don't know. You know, it could be some oligomeric form of the protein. It doesn't have to be the filament at all. And I think the same true of the alpha-synuclein thing. Even with the, you know, experiments with a recombinant protein, we aggregate the protein into filaments. So there are clearly filaments in there, but there's probably also all sorts of other intermediates. And we're injecting the lot. I mean, nobody's so far separated these fractions and injected them separately, and I think that's what one has to do.
It was a question about cell-to-cell spread. I wonder if you could comment briefly on the evidence that these aggregates can actually spread from one cell to another.
Yes, they seem to be able to do that.
Well, what's the evidence?
There is experimental evidence in cultured cells. In our experiments in vivo, and I guess other experiments in vivo too, the evidence is indirect. You could argue, I think it's unlikely, but you could argue that there is no spread, that what spreads to some extent is the extract that's injected, and that the concentration, you know, obviously gets lower and lower further away, and that therefore it takes longer, but it may still be taken up to some extent, and therefore what we're seeing may look like a spread, but it may not be a spread. I would say the best evidence that exists is probably from cultured cells, but there's some evidence by electron microscopy that suggests that it's certainly taken up by cells. Nobody's seen, though, so far as I know, that it's released from cells and taken up by neighboring cells after it's been taken up first by one cell. I think that hasn't been shown. So, yeah, I mean, it's not 100% safe. It's true.
Can you give me a sense from what you're doing with the brain extracts, like compared to synuclein, the tau? The seed for synuclein is itty bitty once it gets in the cell, so very small.
What?
The seed that you get with synuclein is itty bitty. It's tiny. That's all it takes to get a massive accumulation of synuclein in the cell. So, do you have a sense yet what you're seeing with the brain extracts or with tau? even in your in vitro studies, possibly. Same
you
thing.
mean in terms of amounts?
Yeah, what does it take? Same concentration?
I don't know.
Just a tiny seed?
None of this is quantitative. I mean, the synuclein stuff is not quantitative either. The problem is that, again, I think it comes down to the question of what molecular entity it is. Because what you're injecting is a mixture of things. And it could be something in there that's present at a very, very low level, but is incredibly active. So I don't think we can say at this point. Yes. Yes, yeah. So that's another thing.
Hold on. Could you just repeat the question?
Yeah.
Sure. Virginia showed in her science paper that she took the very mature synuclein fibrils and then sonicated them to form PFFs.
Yes.
So
so all the experiments we've done so far, both with the brain extracts and the recombinant protein, we've also sonicated. I think nobody so far, and I think one should do that experiment, is to compare sonicated versus non-sonicated material, because it could be that the sonication is crucial because it creates lots of free ends if the phenomenon is at the level of the fibril.
Yeah, there was a question of mechanism of transport or absorption of PHFs, and we have data that we present here in a poster that PHFs actually can be endocytosed. We have confocal microscopy studies done on it, very careful, to provide us with some proof, let's say, indirect proof that PHFs really can be endocytosed. We don't have yet proof that they can be excluded from the cells. They can be
I think that's,
presented in exosomes. And we think that perhaps we were using a little not exactly good probe to study it, because as we know, Goedert found that expression of exon 2 and 3 sequences in tau are very important for regulation of this exosomes formation and exclusion or induction of this process. So these are the mechanisms we know for the time being. However, we went a little farther with this, and we found that PHFs, when they enter the cell, they provoke really some changes in, they may provoke some changes in proteasome activity. We have now proof in vitro studies, however, not in cells, but in vitro, in cell extracts only, that PHF, incubation with PHFs causes some inhibition of proteasome activity. Right. So this would be, I think, a good mechanism, perhaps, for that spreading phenomena, or one of the mechanisms. Yeah. We don't know about autophagy yet, but we know of proteasome activity.
Because that's another thing about this phenomenon, does it lead to neurodegeneration or not? That's not, I mean, as far as tau goes, that's not so clear at the moment in the animals. But it may have something to do with the relative inefficiency of the methods we have so far. So I think we'll have to wait and see.
One last question. Can you say anything at this point about whether an abnormal conformation, folding conformation of tau can serve as a template to subvert its normal brethren into misfolding?
I think that must be the case. Otherwise, none of these ALZ17 experiments, they shouldn't work.
Right, but no direct evidence for it yet, though.
Direct evidence, it's hard to know. I mean, it's well known that if you, in vitro, if you assemble tau into filaments, monomeric tau, you need a certain amount. If you then take a seed of that material, very small amount, but of aggregated protein, and you add that to the monomer, you need much less to get the monomer to. I mean, as far as I know, what nobody has shown that if you have a seed of aggregated tau, you bind one monomeric molecule to it, then that then changes its conformation and gets integrated. I think it's most likely to happen, but the direct demonstration, as far as I know, hasn't been shown. I mean, I would need to use sort of single molecule techniques, I guess, in order to do that. In principle, it could be done, though.
Okay. All right. Thank you very much, Michel.